Spatial coding in V1. V1 is at the back of the brain. This is the brain looking from the top, and it's also called the primary visual cortex. These are the two eyes, left and right eyes, and the retinal ganglion cells will send their outputs to V1 via an intermediate structure called lateral genetic nucleus. And in these neurons, the recidiv fields in V1 are slightly different, quite different in fact. So this is a schematic showing how different they are. This is the recidiv field in space. It looks like it's oriented horizontally with an on-center horizontally oriented and two flanking inhibitory surround. And this kind of recidiv field can be made, for instance, by such an arrangement done from Hugo and Wiesel's original suggestion. So imagine you have three center surround recidiv fields here, and they are all center surround, but they are lined up horizontally. So the centers add up excitatory for here, and the surround add up inhibitory for here and there. And these are not only kind of recidiv field, they are all kinds. So these are some varieties. For instance, you can be oriented horizontally or vertically, and you can have an on-center here and two flanking surround, or you can have an subfield, you call it subfield or recidiv field, to one side of the recidiv field and the off subfield to the other. You may even have tilted ones. This is tilted about 45 degrees. You can have recidiv field that are smaller, bigger. And so in V1, there's a huge variety of these recidiv fields. Many are tuned to orientation, and some tuned to higher spatial frequency than others, and tuned to different orientation and so on. And some are actually not tuned to orientation, some tuned to color and so on, tuned to motion direction and disparity. Now, if we just focus on spatial coding, we can ask ourselves, can we understand these various types of recidiv field from our efficient coding principle? For example, we have known that efficient coding can be understood from these three conceptual steps: decorrelation, gain control, and multiplexing. And in particular, we've talked about multiplexing actually has many different choices. And for retinal coding, we have chosen a particular choice of U matrix, which is the inverse Fourier transform. Now, can we understand these V1 recidiv field maybe just by a different choice of U? That can be a question to be asked. Meanwhile, you can also ask, why do we need a different version of the efficient spatial coding in V1 if we already have one in the retina? And this question we'll answer later in the book, but not in this video clip. You can use different U transforms to satisfy some additional goals. And one of the goals is to have translation invariance and scale invariance. This means the following. If you have two inputs as x as a function of space x in the image signal, and another image as prime x, and they are related to each other by this relationship, this means this input is a scaled version by a scale alpha of the original input, and a position change maybe by delta x. So this image may be a bit bigger or shifted. And then what you want is the neural responses, so this transform K, for one input S and neural responses for the other input S prime, they should satisfy this relationship. It's the same response, except that for one input, the response is by a different group of neurons. And these neurons are indexed by I plus A means just shifted to another group. And by this shift, and this shift in the neural population is one group of neuron versus another group of neuron. How much shift depends on what is the scale change and what is the translation change, delta x. For example, you can have no scale change, therefore alpha equal to 1, but only a translation of delta x. For example, you can have an image of an apple, and if this apple changes position without changing the distance from your eyes, then the image change will have this kind of change from S to S prime with alpha equal to 1, delta x not equal to 0. And The efficient coding by the retinal ganglion cells can satisfy such translation invariance, because they have the receptor field having the same shape from one cell to another, except their centers, receptor field centers, shift position from one cell to another. So therefore, a shifted apple position will cause this retinal image activate a different group of retinal ganglion cells with their receptor field center location shifted in position corresponding to this shift delta x. However, if your apple moves closer to your eye so that its retinal image becomes bigger with an alpha larger than 1, but its retinal position does not change, so delta x equal to 0, now the retinal ganglion cells cannot respond in a way to make scale invariance, because their receptor fields have roughly the same size and same shape. Therefore, a size change in the retinal image will cause a very complicated change in their population responses that's not describable by such a relationship to keep scale invariance. If your receptor field have these kind of properties, some are smaller, some are bigger, scaled versions of each other, then you might be able to get this scale invariance relationship when you have your retinal image change scale. To ask what kind of U matrix can satisfy our translation and scale invariance property, let's first look at two extreme examples of the U's that we have looked at before to give us some hints. Recall that we have looked at one extreme case when the U matrix is an identity matrix. In this coding transform from decorrelation, gain control, to the U matrix multiplexing. And now if we use a U that's identity matrix rather than the inverse Fourier transform, then all the output units have receptor fields as large as the entire whole image. Each receptor field filter is a Fourier wave. Each neuron has a unique receptor field defined by the wave's frequency, K1, K2, etc. And none of the receptor field is a translated version of another one. One might see that each receptor field is a scaled version of another because the waves differ by their wavelengths, which can be used as a scale. This could help with scale invariance. However, the sizes of the receptor fields do not scale with each other correspondingly, since their sizes are all the same extending over the whole image. Of course, this coding does not satisfy translation invariance at all. On the other hand, if we use the retinal ganglion cell where the U matrix is an inverse Fourier transform, then we will have these receptor fields. They are all centered around for different units is the same shape except translated by the centers of receptor fields, and they will satisfy translation invariance but not scale invariance. Let's list these two extreme cases side by side. One is the identity matrix U, the other is the inverse Fourier transform, in which each column is a Fourier wave from low to high spatial frequencies, K1, K2, etc. So this one, at best, but not quite, only gives scale invariance, and this one only translation invariance. This block diagonal matrix is a compromise to hopefully give both scale and translation invariance. It has the same size, n by n, as the other two. Its non-zero elements are only in the block diagonal submatrices, analogous to this one. But within each submatrix, all the elements are non-zero, just like this one. And each submatrix is a unitary matrix, so that the whole matrix U is unitary as required. And different submatrices cover different spatial frequency bands. And here on the top superscripts are the scale indices S equal to 0, 1, 2, etc. In more detail, let's list out all these frequencies from the lowest to highest, K1 to Kn, and the same list here. And for this retinal filter, we recall that the resistive fields cover the whole frequency range, yeah, from the lowest to highest. Okay. However, now for this multi-scale U matrix, each of these bands has a limited band of frequency. So for instance, let's say for the S band, S equal to 1, the lowest frequency may be here, higher frequency may be there. So this is the frequency range covered for that. And the next S plus 1's band will cover the next frequency range. Here we are considering one-dimensional case, okay, as if the space is one dimension and frequency is just one dimension. And so therefore different scales cover different frequencies. So this one will be even lower frequency and the next one will be higher frequency, so that they can really cover the whole space across these different block diagonal matrices, sub-matrices for the whole U matrix. And we can even calculate the bandwidth within each band. For within each band, you get the highest over lowest frequency ratio, take log 2, that is the bandwidth in octaves. And that gives a fixed alpha for this ratio across these bands. And so with a fixed alpha, this means a fixed width for each of these bands. They have the same width. Yeah. And in V1, this alpha is actually equal to 3. And this gives 1.6 octave for each of these bands. Notice that this frequency axis is on a log scale. And therefore, a fixed alpha here, for example, alpha equal to 3, a fixed alpha here means a fixed width here, even though it means a larger block for the band with a larger mean frequency than another block with a smaller mean frequency, because here it's on the linear scale. The submatrix US, okay, for instance, S equal to 1, takes the same form as this one, except that it has a smaller square size NS, covering only NS out of all N frequencies in this band. And these frequencies are F1, F2, it's these lowest frequency corresponding to here and highest frequency corresponding to highest frequency in this band. Yeah. Now recall that for the retinal filter takes this form. It's Kx. The spatial form is corresponding to a filter for a gain control GK covering all frequencies, summing over all frequencies. Now for this band, it's having the same form, except it just sum over a limited range of frequencies. And similar for here, the receptive field for different neurons are at location x1, x2, all the way to xn, covering the whole visual field. Yeah, and here it's also covering the whole visual field, but has fewer number of neurons. X1, X2, and so on. The number of neurons is this NS. And this is okay because the receptive field is bigger, so it can still span the whole field. The reason this is bigger is because it's having a smaller frequency band. In a one-dimensional visual world, a scale invariant visual world should have this power spectrum. It is 1 over k rather than 1 over k squared for the two-dimensional world. Then if we consider the zero noise limit, high signal to noise, when the gain function should perform whitening, then this should be the gain function. Now let's plug this into this formula for the filter shape, and noting that different bands, different frequency bands for these different submatrices should have the same bandwidth in terms of octaves, then we can derive this relationship between the filter shape in one band and another. So in higher filter band, frequency band, they have the same shape as the lower one, except the spatial extent size is scaled down and the amplitude is scaled up. So therefore, if we use these kind of U matrices for multiplexing, efficient spatial coding is a multi-scale wavelet coding, and this is what we want for scale invariance. This multi-scale wavelet coding property still holds in the two-dimensional visual world. In addition, when alpha is equal to 3, as it is in V1, then in order to keep this U matrix unitary, it is required that there are two different resistive field shapes for each band, so that neighboring neurons alternate between these two shapes for their resistive fields. These two shapes are described by a quadrature phase relationship as seen in experimental data. These and other important details are in the textbook, and you can read them at your own pace to digest further.